Hello viewers, today we will talk about the alternative respiratory pathways in plants. This figure shows the organization of all the different respiratory pathways present in plant mitochondria. As we can see, the respiratory electron transport pathway of plant mitochondria comprises the cytochrome pathway and an alternative pathway. Both the cytochrome and the alternative respiratory pathways start at protein complex 1 when NADH is being oxidized. A proton is transported by the complex 1 to the inner membrane space, whereas 2 electrons are transported within the inner membranes by ubiquinone, which at its reduced state, that is QR, transfers these electrons either to complex 3 or to another protein known as the alternative oxidase or AOX. Ubiquinone is a point at which the reactions can proceed in different ways. That is why it is called the branch point. The cytochrome pathway is present in all living organisms and proceeds when complex 3 pulls out a proton from the mitochondrial matrix to the intermembrane space. The electrons are received by cytochrome C, which spreads up to the outer side of the inner membrane towards protein complex 4, which then pulls out another proton similar to complexes 1 and 3 and transports the electrons back to the inner domain of the mitochondria. As a result, oxygen is consumed with proton and the 2 electrons to produce water. Electron transfers through the cytochrome pathway is coupled with ATP synthesis and is inhibited by cyanide, azide and carbon dioxide. The alternative respiratory pathway is a feature typical of plants, algae, fungi and to some extent protozoa. This type of respiration is brought about by the protein alternative oxidase or AOX, which is a dimer in its inactive form, that is oxidized state. Oxygen is consumed and through a reaction with electrons transported to AOX and proton, water is produced. Thus, both the respiratory pathways transfer protons to the intermembrane space, transport a couple of electrons and consume oxygen to produce water. However, the difference between the two is that the cytochrome pathway transfers 2 additional protons from the mitochondrial matrix to the intermembrane space and thereby leads to a greater proton gradient. Electron flow from ubiquinone is through the alternative pathway, which is non-phosphorylating, cyanide resistant and can yield only about 1/3 of ATP compared to that generated by the cytochrome pathway. The alternative pathway is inhibited by salicyl hydroxamic acid or SHAM and N-propyl gallate. AOX plays an important role in the integration of carbon metabolism and electron transport, besides having a role in specific cellular and developmental processes. Two other components of the alternative pathway are external rotenone insensitive NADH or NADPH dehydrogenases, in short NDEX, that can accept electrons directly from NADH or NADPH produced in the cytosol. And the other one is the internal rotenone insensitive NADH or NADPH dehydrogenases or NDIN that exist on the matrix side of the membrane. AOX for a long time has been considered to act as an overflow mechanism. This is energy overflow, with the exception or modification that it can compete for and share electrons with cytochrome C oxidase. The overflow model associated with AOX appears to strike a balance between carbon metabolism and electron transport. This is because a metabolic condition that causes accumulation of either reduced ubiquinone or mitochondrial pyridine nucleotides or pyruvate or citrate has the potential to increase electron flow to the alternative pathway. The mitochondrial electron transport adjusts its capacity through the alternative pathway, and this has two important implications for plant metabolism. First, if cells need a large amount of carbon sources without a high ATP demand, then AOX facilitates operation of the TCA cycle by consuming NADH. Secondly, AOX prevents over-reduction of respiratory chain components that might result in the production of harmful reactive oxygen species or ROS. Thus, AOX plays an important role in the avoidance of cell damage by ROS. This is imperative in vitro studies using tobacco AOX mutants, where cells overexpressing AOX contained half as much ROS as control cells, whereas cells with reduced AOX expression due to antisensing contained 5 times more ROS than control cells. Thus, AOX plays a role in preventing the formation of oxygen-free radicals. Ubiquinone is a common substrate for both the cytochrome and the alternative respiratory pathway. A high reduction state of the ubiquinone pool, that is a high QR by QT value, is a feature when the cytochrome pathway is inhibited or restricted and promotes oxygen free radical formation. Respiration via the alternative pathway can help maintain QR by QT value at a low level by removing electron from reduced ubiquinone. Nitric oxide or NO is a key signaling molecule that has been reported to be involved in a wide range of processes required for plant growth, development and reproduction, such as seed germination, root growth, leaf expansion and senescence, the establishment of symbiotic interactions, flowering and fruit ripening. NO also mediates adaptive plant responses to various environmental stresses. For example, NO is required for plant disease resistance and tolerance to drought, oxygen deficiency and other abiotic stresses. The diverse action of NO in biological systems reflects its physicochemical properties. NO is a gaseous free radical, highly mobile in cellular systems, diffusing freely in both aqueous and lipid environments with a relatively long half-life, approximately 5 seconds when compared to other radicals. Despite the apparently simple structure of NO, its complex chemical properties in biological systems allow the formation of multiple secondary and tertiary products known collectively as reactive nitrogen species or RNS. NO and each of these RNS can interact with the redox centers of various proteins and transiently or permanently alter their functions and or activities. For example, NO reacts with Fe2 plus in heme or the iron sulfur centers of proteins to form nitrosyl complexes. The numerous NO responsive genes and promoters revealed by large-scale transcriptome analysis have demonstrated significant role for NO in the control of gene expression in plants. NO modulation of transcript levels of genes involved in various signal transduction pathways such as those involved in disease resistance, stress responses and basic metabolism has been demonstrated. Recent studies have demonstrated that mitochondria may play a central role in various NO-mediated effects in plants because the synthesis and degradation of NO are developed within these organelles. Additionally, NO and its derivatives have multiple effects on mitochondrial bioenergetics, thereby affecting the overall cell physiology. Therefore, as has been shown in mammals, the interaction between NO and the mitochondrial respiratory chain may mediate the biological effects of NO in plants. The uncoupling pathways for electron transport have physiological significance for the control of NO levels. This diagram is a schematic model for the maintenance of NO homeostasis by plant mitochondria. As we can see, nitrate is reduced to nitrite by cytosolic nitrate reductase or NR. Nitrite is then reduced to NO by cytochrome C oxidase or COX or complex 3 of mitochondrial respiratory chain. At physiological levels, NO causes reversible inhibition of COX and can also lead to S-nitrosylation of complex 1. The resulting restriction of electron flux through the cytochrome pathways stimulates production of superoxide by index and complex 3. These enzymes then contribute to NO degradation because NO promptly reacts with superoxide to produce peroxynitrite. Conversely, AOX allows mitochondrial electron flow in the presence of NO and decreases electron leakage and NO consumption. Peroxynitrite can be metabolized back to nitrite by peroxyredoxins, and NO can also be metabolized to nitrate by cytosolic class 1 non-symbiotic hemoglobins, closing the cycle. Thus, alternative pathways for electron flow in the mitochondrial respiratory chain may play a major role in the control of NO homeostasis. Physiological levels of NO produced under aerobic conditions restrain the cytochrome pathway, which causes over-reduction of the electron carriers. Consequently, increased production of ROS occurs at the level of index and complex 3. The consumption of both radicals by the spontaneous reaction of NO with superoxide to generate peroxynitrite helps to minimize ROS and NO levels and allows the recovery of oxygen consumption by mitochondria. Insensitive to NO, AOX enables electron flow by the respiratory chain in the presence of NO, thus limiting ROS production, NO degradation and peroxynitrite accumulation. Therefore, a role for index and AOX in maintaining NO homeostasis in plant cells seems quite possible. Steady state levels of NO are also controlled by the rate of synthesis of this radical. Although various cellular sources of NO have been proposed, the reduction of nitrite by the mitochondrial respiratory chain has emerged as an important mechanism for NO production in plants. This mechanism is enhanced as oxygen concentration decreases, and COX is the main candidate for this activity with cytosolic NR providing nitrite. When the production of NO is overwhelmed and high levels of RNS accumulate, the mitochondrial respiratory chain may be affected at multiple sites, which may cause the irreversible inhibition of respiration and lead to cell death. In this scenario, the tolerance of plants to different types of stresses may be related to their ability to control steady state levels of NO, in which the induction of alternative respiratory pathways could play a central role. When electrons pass to the alternative pathway from the ubiquinone pool, two sites of proton pumping, namely at complexes 3 and 4, are bypassed. Because there is no energy conservation site in the alternative pathway between ubiquinone and oxygen, the free energy that would normally be conserved as ATP is lost as heat when electrons are shunted through the alternative pathway. One example of the functional usefulness of this thermogenesis by AOX is its activity during floral development in certain members of the RAC, the arum family, for example, the voodoo lily. Just before pollination, tissues of the club-like inflorescence called the appendix, which bears male and female flowers, exhibit a dramatic increase in the rate of respiration via the alternative pathway. As a result, the temperature of the upper appendix increases by as much as 25 degrees centigrade over the ambient temperature for a period of about 7 hours. During this extraordinary burst of heat production, certain amines, indoles, and terpenes are volatilized, and the plant therefore gives off a putrid odor that attracts insect pollinators. Salicylic acid, a phenolic compound related to aspirin, has been identified as the chemical signal responsible for initiating this thermogenic event in voodoo lily. Carbon fluxes through plant mitochondria in the light. Mitochondria of C3 plant that lives in the light support oxidation of the high fluxes of photorespiratory glycine, which is synthesized via the glycolate pathway as a consequence of the oxygenase reaction of Rubisco. The oxidation of respiratory substrates, although partially inhibited, proceeds simultaneously with the photorespiratory glycine oxidation. The alternative non-coupled pathways of the mitochondrial electron transport and the reactions associated with these pathways play important roles in the maintenance of high photorespiratory flux. High rates of glycine oxidation are possible via the kinetic mechanisms leading to the increased capacity of the mitochondrial electron transport chain under intensive glycine oxidation via switching to non-coupled pathways. Raising NADH due to the GDC or glycine decarboxylase complex reaction not only engages the non-coupled pathways but also results in the intensification of the malate and citrate mitochondrial valves. This is the scheme of the GDC reactions catalyzed by its different proteins with links to metabolic processes. As we can see, P protein is involved in decarboxylation, L protein in NAD+ reduction, and T protein in release of ammonia. Carbon dioxide is equilibrated by carbonic anhydrase with bicarbonate, which is exported to the cytosol. Ammonia is produced and protonated to ammonium, which is exported and used in chloroplast. NADH is equilibrated by the mitochondrial malate dehydrogenase. Malate is exported to the cytosol where it is equilibrated by cytosolic malate dehydrogenase. Cytosolic NADH can be oxidized by NDEX, mitochondrial NADH by complex 1 and endine. Mitochondrial NADPH formed in the non-proton pumping transhydrogenase reaction is oxidized by endine. The electrons from the ubiquinone pool are transported to oxygen either via the cytochrome pathway, that is complexes 3 and 4, or via the alternative oxidase. Nutrient deficiency or changes in soil nutrient composition is often observed in nature and may inhibit growth and have a severe impact on crop yield. Uptake and assimilation of nutrients requires a substantial amount of energy in the form of ATP and thus reduces the plant resources. Therefore, adaptation to variations in nutrient supply requires changes in plant metabolism. Now, plant metabolism is generally flexible because of the presence of alternative respiratory pathways that facilitate adaptation and continuation of growth in a changing environment. The main source of energy for ion uptake and transport and assimilation is derived from respiration. The flexibility of respiratory pathways enables plant growth and development in different nutrient conditions. Most nutritional stresses in an increased reduction state of cells or individual compartments. The limited availability of phosphate and consequently restricted ATP synthesis influence energy metabolism, activating bypasses and omitting ATP phosphate dependent steps in the glycolytic pathway and the mitochondrial electron transport chain. Low nitrogen supply due to a decreased energy demand required for biosynthesis, ion uptake and transport results in the general suppression of glycolytic and cytochrome pathways in the cytosol and mitochondria respectively, but activates mitochondrial AOX. Lack of sulfur in the growth medium of beans leads to suppression of mitochondrial complex 1 and activation of endine but not NDEX. Thus, due to the branch structure of mitochondrial electron transport chain possessing endine, NDEX and AOX, the mitochondria are important players in the regulation of oxidation reduction homeostasis during nutrient stress. AOX activity was found to be higher in sun species than in shade species. Alocasia odora, a shade-loving plant, regulates its respiratory capacity by making changes in the mitochondrial number in leaves when subjected to growth under varied light regimes. It maintained a high AOX activity and capacity whose activity was controlled by keeping AOX protein as inactivated under low light. However, this inactivated oxidized dimer was converted to a reduced active form once the plants were shifted to high light conditions. There is growing evidence that AOX plays an important role in balancing photosynthesis and respiratory metabolism under high light stress. The AOX pathway protects plants from the effects of photoinhibition. The NADPH produced in chloroplasts and transported into mitochondria via various shuttles such as malate oxaloacetate shuttle is oxidized by mitochondrial AOX. AOX does this without being restricted by a proton gradient across the mitochondrial membrane or the ATP-ADP ratio as shown in rumex leaves. AOX also imparts protection against photooxidation damage by regulating ROS production, which stems from photosynthetic electron transport. ROS production increases with increasing light sensitivity. Excess ROS causes photooxidation damage to photosynthetic apparatus. AOX suppresses ROS production and maintains the photosynthetic electron transport chain in an oxidized state during stress conditions. So viewers, we have discussed today the details of the alternative respiratory pathways and their significances in plant. They have important effects such as recovering the components of electron transport chain from highly reduced state. They also play a very important role in thermogenesis and controlling reactive oxygen and nitrogen species under different stress conditions. It can also help adaptation of the plant in high light stress. Thanks for listening.